This is the first video of the ionotropic receptors first lecture. In this introductory video, we will discuss very briefly the different types of synapses that we can find in the brain, different neurotransmitters, types, and their functions, and very briefly the different types of neurotransmitter receptors. So, as many of you know already, neurons communicate with each other via synapses. And there are two main types of synapses. There are electrical synapses and chemical synapses. And electrical synapses contain a large number of gap junction channels that communicate the pre- and postsynaptic cell. When an action potential arrives to the presynaptic terminal, the electrical signal travels through the gap junction channel, inducing an inward current in the postsynaptic cell, and therefore causing a depolarization of the postsynaptic cell. This type of synapses can actually be bidirectional. In contrast, in the chemical synapses, when an action potential arrives to the presynaptic terminal, it triggers the fusion of vesicles containing neurotransmitters with the membrane of the presynaptic terminal, releasing neurotransmitters to the extracellular space. And then neurotransmitters bind to specific ionotropic receptors present in the postsynaptic terminal. And activation of these receptors leads to changes in the membrane potential of the postsynaptic cell. In this case, an electrical signal is transduced into a chemical signal that is then transformed back into an electrical signal via activation of the ionotropic receptors. And these synapses are unidirectional. So for the next 3 lectures, we will focus on studying chemical synapses, specifically how different ionotropic receptors work and transform chemical signals from neurotransmitters into electrical signals. So let's start with what are neurotransmitters and what kind of neurotransmitters we can find in the brain. Well, neurotransmitters are body's chemical messengers composed by simple molecules, derivations of amino acids, or even small peptides that are packaged and stored locally into small vesicles. This type of amino acids are stored in vesicles that are around 40 or 50 nanometers. The first neurotransmitter to be discovered was a small molecule called acetylcholine. It plays a major role in the peripheral nervous system and have roles in maintaining and modulation both motor and cognitive function. Glutamate, on the other side, is an amino acid with a primary excitatory function. Conversely, a major inhibitory neurotransmitter is the gamma aminobutyric acid, or GABA. And another inhibitory neurotransmitter is the amino acid called glycine. Many other neuromodulators are monoamines, including catecholamines such as epinephrine or adrenaline and noradrenaline. These are monoamines and work on the activity of various organs in the body to control things like blood pressure or heart rate, liver function, and many other functions. And then we can also find in this group dopamine, which is involved in many functions, including motor control, reward, and reinforcement and motivation. The neurons that use serotonin, which is another monoamine, functions in sleep or memory, appetite, mood, and others. Then another neurotransmitter from this group is histamine, which is the last of the major monoamines and plays a role in metabolism, temperature control, regulating various hormones, and controlling the sleep-wake cycle, among other functions. And then on the other hand, we have neuropeptides, which are small protein-like molecules or peptides found in Larger vesicles, as we can see here, there are about more than 100 nanometers dense core vesicles. And few examples of neuropeptides are enkephalin, which is a pentapeptide involved in regulating nociception in the body, or somatostatin, which is a peptide hormone that regulates the endocrine system. So, important things are the many neuropeptides are also co-released with other small molecule neurotransmitters. And one major difference is that peptides are not recycled back into the cell once they're secreted, unlike many conventional neurotransmitters such as glutamate or dopamine or serotonin. And another difference is that after secretion, peptides are modified by extracellular peptidases, activating them or sometimes inactivating their function. In this slide, you have examples of several neurotransmitters, examples of what they are working on and what their structures look like. You definitely do not need to memorize any of this, but this is just an illustration to see how different neurotransmitters look like. And I want to emphasize 2 points here that I'm pretty sure you already know, but just to make sure. One is that a neuron can release more than one neurotransmitter, and that neurotransmitters are ineffective without the appropriate postsynaptic receptor. So when the neurotransmitter is released by a presynaptic terminal, it needs to find the appropriate receptors to exert their function. So when a neurotransmitter is released, it then needs to interact with its appropriate receptor. And then upon binding, it leads to the formation of a particular signal, which through a series of different reactions result in some biological effects. For example, a signal can tell the DNA to stop replicating, or it can tell the DNA to start producing more proteins, or it can actually bind and induce opening or activation of other receptors. So cells in our body have lots and lots of different receptors, which when stimulated can produce very unique responses. The receptors that have the most therapeutic relevance can be divided into 4 different types. One is the ligand-gated ion channels or ionotropic receptors, which will be the focus of this week's session. Then there are G protein coupled receptors or GPCRs, or also known metabotropic receptors, which will be seen here briefly, but will be covered in more detail by other professors in following sessions. Then we have also enzyme-linked receptors and intracellular receptors. And we will talk a little bit about each of these receptors here in this introductory video. And as I mentioned, ligand-gated ion channels will be seen in more detail in the following videos. So here there is an overview of a ligand-gated ion channel. These are the easiest one to describe. And just a quick reminder, when we refer to a ligand can be some molecule or it can be also an ion. So when this channel it's bound by a ligand in its binding site, then the ligand executes a conformational change that causes the channel to open very briefly. And this allows ions such as sodium, potassium, chloride, or calcium to pass through the membrane. And that's all they do. So in this scheme on the right side, you can see an overview of a representative subunit of an ionotropic receptor, in which you can see the transmembrane domains that are forming the pore and are considered the membranes that form the channel.
Then in the extracellular space, there are two important domains, the ATD or amino terminal domain, which is usually part of these receptors being interacting with other potential proteins. And then the LBD or ligand binding domain, which is the place where the ligand binds. And then some subunits may have additional domains, such as the CTD or carboxy terminal domain, that may have very complex structures that allow them to interact with other proteins in the intracellular space. So next we have the metabotropic receptors, also known as G protein coupled receptors or GPCRs. These are receptors that involve a complex of proteins that result in metabolic changes within the cell, and they're characterized by the production of a second messenger that acts from the inside of the cell to open an ion channel and or elicit other intracellular events. And we will see some examples of that. These receptors are considered a 7-transmembrane type receptor because their transmembrane domain crosses the membrane 7 times, having 7 alpha helices embedded in their membranes. And they are also formed by a G protein. And this G protein has 3 different subunits, alpha, beta, and gamma, with different and specific functions that we will see in the next slide. And then these G protein coupled receptors have an effector protein that will be either activated or inactivated depending on the composition of these G subunits.
The G proteins associated to metabotropic receptors are heterometic protein complex composed by 3 subunits, alpha, beta, and gamma. In this scheme, you can see how these G proteins are activated in response to a neurotransmitter. So upon ligand binding, there is a conformational change that facilitates the exchange between GDP to GTP. In this process, the alpha subunit of the heteromeric G protein complex dissociates from the complex and it moves away from the receptor to activate the effector protein. Although this scheme doesn't represent what I'm going to say, the beta and gamma can also form a complex that can also dissociate from the receptor to elicit other functions. And then once the alpha subunit has executed its activity, the GTP is hydrolyzed again back to the GDP form, and the entire complex reassembles again. This is a scheme of how metabotropic receptors work in general. There are different variations to this activation scheme that we will see in the next slides. But in general, metabotropic receptors work or they get activated when the neurotransmitter binds to the extracellular space to this N-terminus part of the metabotropic receptors. Binding of the neurotransmitter causes a conformational change in the receptor and activates a G protein that is located in the intracellular space and is associated with the C-terminus part of the receptor protein. This G protein is a GTPase hydrolase that requires the energy from the GTP to physically move away from the receptor and activates an effector protein. And in this case, it's an enzyme that catalyzes the generation of a new molecule or second messenger that acts in the intracellular space to mediate a signal that causes the ion channel to open, but also produces other cell responses that can modify protein phosphorylations and transcription. So, metabotropic receptors can interact and regulate the function of ionic channels by either opening or closing ion channels. Or they can activate different effector proteins, initiating different intercellular pathways, such as those mediated by the adenylate cyclase or the phospholipase C. And these have different molecular mechanisms to control ion channel function and modulate transcription.
And in this slide, you can find a more specific example of how the both AC and PLC pathways are activated through G protein coupled receptors. And I'm not going to go into the details of these specific pathways because, as I said, you will be seeing this in other lectures in this class. So now let's move on to the enzyme linked receptors. These receptors, just like G protein receptors, have an extracellular binding site where ligand, typically a hormone or a growth factor, can attach and then stimulates an enzymatic activity inside the cell. Most enzyme-linked receptors are tyrosine kinase, which simply means that they display a kinase activity in which there is a tyrosine, an amino acid tyrosine, involved in the process. And the way this works is that when the ligand binds to two of these receptors, it causes a conformational change that results in the aggregation of these two receptors and dimerization. And once the dimer is formed, the tyrosine regions get activated here. And it causes ATP to become ADP, which results also in an autophosphorylation of the receptor. So now, each type of tyrosine picks a phosphol group. And different inactive intracellular residues come up and attach themselves to phosphorylated tyrosine, such as this type of molecules. This, in turn, cause a conformational change in these attached new proteins, ultimately leading to a cascade of activations that produces a particular cellular response. And next, the last type of receptors is, unlike the others, an intracellular receptor. This receptor is located entirely inside the cell rather than on the surface or on the membrane of the cells. And therefore, the ligand has to first cross the lipid membrane. And then once inside, it can then bind to the receptor. So now the activated ligand receptor complex can move into different compartments, mostly the nucleus, to bind DNA and regulate gene expression, ultimately leading to synthesis of specific proteins. So this is an overview of the different type of receptors. In the next two other videos, we will cover in more detail the structure and function of ionotropic receptors.
And finally, as usual, I leave you with a short quiz where you can revise some of the most important concepts that were covered in this introductory video.